Knowledge portals are open-access web resources that help researchers access and explore genetic and genomic data for complex diseases. They aggregate, harmonize, and display this information so that researchers can identify the genes involved in a disease. Knowing which genes have roles in disease will help us understand the disease, ultimately speeding up the identification of new drug targets and allowing us to better predict and address disease risk. Right now, there are 5 knowledge portals for type 2 diabetes, heart disease, stroke, sleep disorders, and amyotrophic lateral sclerosis, or ALS. Let's focus on the portals for cardiometabolic diseases since they all share a similar format. We'll demonstrate the portals using the Type 2 Diabetes Knowledge Portal. It was the flagship portal, the first to be developed, supported by the Accelerating Medicines Partnership in Type 2 Diabetes. The knowledge portals aggregate both summary statistics and individual-level data from genome-wide association, exome chip, exome sequencing, and whole genome sequencing studies. You can find all the details on the data page. The knowledge portals have 3 major entry points to these results. If you have a gene, genomic region, or variant of interest, you can find more information using the Explore Data search box. Searching with a gene name or coordinates of a region takes you to a gene or region page, respectively, that summarizes what is known about genetic associations and epigenomic marks within that region. The top of this page has a traffic light symbol that shows you at a glance whether there are strong associations in the region, a green light, suggestive, less strong associations, a yellow light, or no associations, a red light, within the datasets in the portal. The rest of the page lets you drill down to the fine details of the associations and epigenomics of the region, and we'll cover it in more detail in an upcoming video. Searching with a variant ID takes you to a variant page. The top section of the page, Associations at a Glance, summarizes the associations of that variant across all of the datasets and phenotypes in the portal, or across a large set of phenotypes from the UK Biobank. The forest plot also displays all associations for a variant and plots effect size and direction in addition to p-value. There is lots more on the variant page, but we'll cover that in a future video. The second major entry point to the portals is the interactive Manhattan plot. To find it, click on the Analysis Modules link on the homepage. This tool lets you browse associations across the whole genome for any phenotype and dataset. Choose a phenotype to see a Manhattan plot. The top associations are listed in a table below the plot. If there is more than one dataset that includes associations for a phenotype, the plot for the largest dataset is shown by default, and you can choose other datasets using the pull-down menu. Some datasets may be clumped by measures of linkage disequilibrium, or LD. You can choose an R-squared threshold and redraw the plot and gene table to reduce the number of redundant signals caused by genetically linked variants. Finally, the third major way to explore the results in a knowledge portal is to use the Variant Finder tool, which is also found on the Analysis Modules page. You can specify multiple search criteria and retrieve the set of variants that answer all of those criteria. For example, variants that are associated with both lower risk of T2D and higher BMI. That's a brief overview of how you can access the results in the knowledge portals. Upcoming videos will have more information about the displays and tools, and we're continually adding new data and features, so check back often for updates.